everybody, and welcome back to the Lead MA. My name is Diana Batista, and I am your host. Today we have an exciting topic to cover in the world of specimen collection, focusing on the crucial concept of order of draw. Mastering this technique is essential for medical assistants like us to ensure accurate and reliable test results. So let's get started. Before we jump into the specifics, let's understand what the order of draw is and why it's so important. The order of draw refers to the sequence in which we collect blood and to different tubes during the venipuncture. Each tube contains specific additives, or no additives at all, designed to preserve the integrity of various analytes that are assessed during the lab portion. Following the correct order ensures that the samples are not contaminated and the test results are reliable. The order of draw typically consists of different colored tubes, each representing a specific additive or the lack thereof. So let's break it down. Starting off our order of draw, we have the yellow tube, which contains sodium polyanethyl sulfonate, SPS, or acid citrate dextrose, which is also known as ACD. This tube is primarily used for blood cultures. Moving on, we have the light blue tube, which contains sodium citrate. It is used for coagulation studies such as prothrombin time and partial thromboplastin time, also known as PTT. The red tube is the most common one you'll encounter. It doesn't contain any additive at all and is used for collecting serum or blood clots for various tests, including blood chemistry, serology, and blood bank. Red, gray, or gold tubes, also known as tiger tops, are also known as serum separator tubes. This tube contains a gel separator and it is used for chemistry tests and blood bank. Following the gold, red, gray tube, we have the green tube, which contains heparin as an additive, and it is used for tests like arterial blood gases, ABGs, electrolyte panels, and ammonia levels. Now let's move on to the lavender tube. It contains the anticoagulant, which I will not pretend I know how to pronounce. It's called EDTA. If you need to look up the full acronym, feel free to do so, and you'll see why I can't pronounce it. This test is typically used for complete blood count and for blood bank testing. Lastly, we have the gray tube, which contains sodium fluoride and potassium oxalate as additives. This tube is used for tests that require any glucose testing, such as blood sugar levels. Now, let's be real. The biggest issue with lab draws isn't necessarily doing the actual sticks, it's memorizing the order of draw. And it can definitely feel a little overwhelming, but fear not. I have a handy mnemonic to help you remember the sequence effortlessly. I used it, and while it's a very weird saying, let's say it together. Yale likes rice, good gravy, green leaves, and grapes. And this is used to represent the colors and the additives. So, yellow is Yale, light blue is likes, red is rice, red, gray, or gold is good gravy, green is green leaves, lavender, and gray is for grapes. Remembering this mnemonic will ensure the proper collection of the tubes in the right order every single time. Now, I get it, it's a weird saying that you would not typically say all the time, but repeat, 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 practice makes perfect. Now that we've covered the order of draw, let's discuss some of the commonly ordered lab tests associated with each tube. Understanding which tests are typically performed with each tube will further enhance your knowledge and efficiency as a medical assistant. Yellow tube, which has the SPS or ACD additive, is commonly used for blood cultures. Light blue tube has the sodium citrate, and the commonly test is PT and PTT, as well as D-dimer. Your red tube, which has no additives, is commonly used for blood chemistry, liver function tests, lipid profile, HIV screening, and next you'll have your red, gray, or gold tube. These are serum separator tubes, and they're commonly used for comprehensive metabolic panel, CMP, glucose testing, cholesterol studies, thyroid stimulating hormone, which is your TSH. Next, we have the green tube. This has this heparin separate additive, and it's used for ABGs, arterial blood gases, electrolyte panels, which is sodium and potassium, and ammonia levels. Your lavender tube has EDTA and is frequently used for complete blood count, CBC, or erythrocyte sedimentation rate, which is your ESR. It's also used for blood typing. Your gray tube has sodium fluoride and potassium oxalate. This is used commonly for fasting blood sugar, lactic acid, and ethanol levels. Remember, these are just some of the examples of your commonly ordered lab tests associated with each tube. Specific testing requirements may vary depending on the healthcare facility and the lab you use, as well as a patient's condition. Ensure that you communicate with the lab that you're frequently using and see what their standard of testing and tube colors are. Now, as we wrap up, let's touch on some important pointers to ensure for successful specimen collection. You always want to ensure that you confirm the patient's identity. Verify the patient's name and date of birth before starting any procedure. Accuracy is crucial to avoid any mix-ups, especially in a high-volume office. Use aseptic technique, maintaining proper hand hygiene, and adhere to sterile techniques during specimen collection. This is going to prevent any contamination. Follow the correct order of draw. As I said before, reach out to your labs and ensure that everything is the way it's supposed to be. Collecting samples in the wrong order can lead to cross-contamination and inaccurate results. Properly label each tube as you're collecting them. Accurate and clear labeling is essential for correct identification of samples. Don't wait until the end. Include the patient's name, date of collection, and preferably the time of draw. Make sure that you're gently mixing the tubes at the time, inverting them. Don't shake, because that can lead to hemolysis. Follow proper disposal protocols that you have in your office for needles, syringes, and different tubes that may have any sharps in them to prevent any needle stick injuries. Also, make sure that you communicate with your lab if you have any difficult time, getting the collection from the patient, and making sure if you have any special handling of the samples, such as they need to be on ice, they need to be frozen, they need to be covered from the light. As medical assistants, we play a crucial role in laboratory testing. Mastering the order of draw and following best practices contributes to optimal patient care. And there you have it, a comprehensive guide to the order of draw and important pointers for successful specimen collection. I hope that this video has been informative and beneficial to you as a medical assistant or somebody aspiring to get into the medical field. Remember, practice makes perfect. Don't be too hard on yourself and always ask the questions that you need. Keep honing in on your skills and never stop learning. If you enjoyed this video, please don't forget to like, comment, share, and subscribe to the Lead MA, as this is a growing channel and I will be providing a lot more valuable content. If you have any questions or suggestions for future topics, feel free to leave them on the comments or message me directly if my information is in the bio. Now, thank you so much for watching, and until next time, stay curious and keep striving for excellence.